If you want to identify a genetic disease, sequence a novel virus, or run next-generation sequencing, you need one specific raw material: pure, intact DNA. But retrieving that DNA is a challenge of scale. The genetic code is incredibly fragile, yet it is physically encased within the dense, multi-layered environment of the nucleus. We cannot simply crush or blend the cell to get it out. Applying raw physical force would shear the delicate, microscopic strands of DNA into useless fragments. Isolating DNA is a highly precise chemical rescue mission. We have to systematically dismantle the cell structure without destroying the valuable cargo it holds inside. To breach the cell, scientists deploy a targeted mixture of chemicals known as a lysis buffer. The first barrier this liquid hits is the cell membrane. This outer wall is constructed almost entirely of tightly packed fats, or lipids. The lysis buffer breaks this wall using sodium dodecyl sulfate, or SDS. It wedges directly into the fat layer, bursting the cell. Once exposed, the DNA is highly vulnerable. A sudden shift in pH would cause the strands to break instantly. To prevent this, the buffer contains a chemical called Tris-HCl. It acts as a molecular shock absorber, instantly creating and maintaining a highly stable pH environment around the DNA. Breaching the cell requires these exact opposing actions simultaneously, aggressively destroying the protective lipid wall, while immediately deploying a chemical shield to stabilize the structure inside. With the outer wall compromised, the DNA faces an immediate internal threat. Cells naturally contain defensive enzymes called DNases, which actively hunt down and chop up loose DNA. These molecular scissors have a strict operational requirement. They cannot function unless they physically bind to free-floating magnesium ions. When they lock onto magnesium, they activate. We stop them by deploying a specific chemical countermeasure called EDTA. It acts like a chemical claw, aggressively hunting down the magnesium ions and trapping them. By starving the enzymes of the magnesium they need to operate, EDTA effectively shuts down the cell's internal defense systems, ensuring the DNA isn't digested by its own environment. Deep within the nucleus, one final biological barrier remains. The DNA is tightly coiled and knotted around structural scaffolding known as histone proteins. To loosen this grip, we place the sample into a heat block and raise the ambient temperature to precisely 56 degrees Celsius. At this exact temperature, a specialized enzyme called Proteinase K reaches its maximum efficiency. We add Proteinase K to the sample, where it actively digests the blocking histone proteins. With the scaffolding gone, the dense knots of DNA finally unspool into long, continuous strands. The precise combination of heat and enzymatic digestion completes the demolition. The highly structured cellular organism is now a chaotic, free-floating soup of raw biological materials. Now we face a pure logistics problem, physically extracting the microscopic DNA molecules from a chaotic liquid filled with dead proteins and dissolved fats. To filter this mixture, we use a spin column containing a silica membrane. Chaotic salts force the negatively charged DNA to chemically glue itself to the silica. When spun, the biological debris flows straight through, leaving only DNA caught inside. The DNA is trapped, but it's still covered in leftover salts and stray proteins. We clear this using a series of wash buffers formulated with a high concentration of ethanol. The ethanol washes over the membrane, pulling away the microscopic debris. Because DNA cannot dissolve in ethanol, its chemical bond to the silica remains totally secure during the wash. By manipulating these specific chemical binding affinities, we physically trap the genetic code on the filter while flushing all remaining biological waste out of the bottom of the tube. The DNA is now completely isolated and pure, but it is permanently stuck to the silica membrane. To release the strands, we apply a specific low-salt liquid, an elution buffer, or in some cases, highly purified water. This final liquid intentionally lacks salt. When it hits the filter, the chemical environment abruptly shifts, causing the DNA to break its bond and easily through the pores of the filter and raining down into the final collection tube. Dismantling the cell and isolating its instructions provides the high-purity input that modern sequencers require. Every downstream genetic insight begins with this specific series of chemical reactions.